From the American Neurological Association, I'm Adeline Goss. This is ANA Investigates. The brain and its vasculature used to be considered separate domains, but in the last decade, we've learned more about the interplay between brain cells and vascular cells. Pathological studies are revealing a surprising degree of overlap between cerebrovascular disease and neurodegenerative disease. And all of this has led to the coining of a new term, the neurovasculome. Our guest today was an author of a recent scientific statement about the neurovasculome in the journal Stroke, Dr. Eric Smith. Dr. Smith is a professor of neurology, radiology, and community health sciences at the University of Calgary and the holder of the endowed Katthy Taylor Chair in Vascular Dementia. He was interviewed about the neurovasculome by Dr. Christoph Stretz, Vascular and Critical Care neurologist at the Warren Alpert Medical School of Brown University. Here's Christoph.
Welcome to the show this afternoon, Eric. We really appreciate you joining us today for our podcast.
Thank you.
I think to start us off, I would like to ask you, can you describe what we mean by the term neurovasculome, and how do you differentiate that from another term that I came across, the neurovascular unit?
Yeah, great question. And the neurovasculome is a relatively newer term, and our ability to talk about it has been enhanced by developments in the characterization of vascular cells that allow us to fully appreciate the complexity and diversity of the different cells in the vascular tree that supplies the brain. So the neurovasculome refers to all the different cell types that are involved in delivery of blood and the exit of blood from the brain. So that includes the cells comprising the arteries, the capillaries, the veins, and the closely associated cells, all the way from the heart and the aorta to the brain and back. So the neurovasculome cells are essential for functioning of the neurovascular unit, which is a term that refers to the collaboration between the vascular cells, the neurons, and the supporting cells like the astrocytes and the microglia that function in a highly integrated manner to enable brain output. The neurovascular unit varies across the different levels of the vascular tree, depending on which vascular cells are present, but at every level, there are vascular cells, neurons, astrocytes, and microglia working together to produce brain output. And so that is what the neurovascular unit refers to.
Thank you very much for this comprehensive overview over those definitions. Could you describe to our audience some of the key functions of the neurovasculome and mechanisms how it achieves them?
Yeah, neurovasculome cells have to be all functioning optimally to enable the neurovascular unit to function. So one of the most important functions is perhaps the most obvious. They provide a physical conduit for blood, which is delivering oxygen and glucose, and other energy substrates to the brain for the brain to consume energy and function properly. In fact, we know in the early stages of neurodegeneration, we see some alteration in this function. We see some leakiness of the vasculature at the capillary level that can be detected with quantitative neuroimaging. And then, of course, if delivery of blood flow is completely interrupted, we see ischemia leading to infarction, right, if ischemia is not restored. And so that's what happens when you have ischemic stroke. And then gross disruption of the vascular tree, allowing exit of blood from the system, leads to forms of hemorrhagic stroke, intercerebral hemorrhage, or subarachnoid hemorrhage. And those are destructive events for the brain. So that's been recognized for a long time. But of course, the vasculature does many other things. It's a highly adaptive functioning organ system, not merely a passive set of pipes. So one of the things it must do is it has to regulate blood flow to different parts of the brain as those parts of the brain are functioning. So when certain parts of the brain are working harder, blood flow is increased to those parts of the brain. This phenomenon can be imaged and is the basis of functional MRI, which we can use to identify through changes in blood flow and consequently oxygenation. We can identify regions where blood flow delivery is enhanced in parts of the brain that are working harder. You can use that to, for example, give a task to a patient And then by imaging the blood oxygen level dependent signal from fMRI, you can see which parts of the brain are more active. So that's another important function that involves a lot of communication. And we know now that this communication to enhance blood flow to parts of the brain that are more metabolically active also involves essentialist signaling through the astrocytes as well. Another important function is through clearance of wastes. So the vascular system delivers energy substrate, but it also carries away waste products, including fairly strong evidence now that it plays a critical role for clearance of the beta peptides that, if not properly cleared, will aggregate to form dimers, trimers, oligomers, and eventually deposit in the brain as neuritic plaques, which is the hallmark of Alzheimer's disease. So these are some of the diversity of functions that the vascular system provides, and it needs the integrated functioning of diverse cell types in the neurovasculome to accomplish these tasks.
You mentioned waste products that are removed, and one term I came across in your statement are perivascular spaces. And I was wondering if you could briefly elaborate on what our understanding is, in particular with Alzheimer's disease, how they are connected.
Yeah, there are physiological spaces around the arteries and arterioles and also veins in the brain called perivascular spaces. They're line spaces and they're actual existing spaces, not virtual spaces, essentially filled with cerebrospinal fluid. They disappear at some point when you get down to the level of the smaller arterioles, or at the kind of the transitioning between arteries and arterioles. The spaces disappear, and at that point the vessels are just surrounded immediately by the brain parenchyma. These spaces have been potentially implicated in this waste clearance phenomenon. And it's important to understand there's two theories and possible mechanisms, each of them with some experimental support on the direction of clearance of waste from the brain. One of these two pathways is called the IPAD, or the intramural periarterial drainage pathway. And this posits that solutes and fluid move out of the brain along the basement membrane of arteries, which are closely anatomically located to these perivascular arterial spaces, and then exit the brain that way. The second pathway that's been proposed, for which there's also experimental support, is called the glymphatic pathway. In this pathway, bulk fluid moves and exits from the brain in the opposite direction along the venules and veins, and then out into the venous system. And this has been proposed to be mediated by aquaporin pumps on the astrocytes as providing the motive force. The motive force for the first pathway, intramural periarterial drainage, is not entirely clear, but may have something to do with the arterial pulsations, because these small arteries are pulsating with each beat of systole. It may be that both pathways are active to some extent. We don't fully know, and this is one of the areas where we need more investigation and for research. The perivascular spaces, the larger of them are visible on brain MRI. So this is something that we can see in humans, particularly on a T2-weighted scan. And there are grading systems that have been proposed, implemented, and shown to be reliable to grade the number of visible spaces. As mentioned earlier, they're anatomic spaces, so they're physiological. Everybody has them. But it appears that in some people, as they get older, they become wider, and then more of them become visible on T2-weighted imaging. The grading scales rely on counting the number of visible spaces in the most affected hemisphere. The most commonly used scale, developed by Joanna Wardlaw and colleagues, counts them in the basal ganglia as well as in the centrum semiovale white matter. In the vascular condition cerebral amyloid angiopathy, you see that the spaces, particularly the ones in the centrum semiovale, are much more visible. Cerebral amyloid angiopathy, or CAA, is caused by deposition of beta amyloid in the walls of small arteries, arterioles, in the leptomeninges and cortex. And it's been hypothesized that this widening of the spaces in cerebral amyloid angiopathy may have something to do with the process that's leading to deposition of the vascular amyloid in the first place. Like, it may reflect slow movement of fluid through these spaces. So the number of visible spaces may be a surrogate for clearance from the brain. They're also seen in patients with non-cerebral amyloid angiopathy, small vessel diseases like deep perforator arteriopathy from hypertension, although in that case you see the perivascular spaces are more widened in the basal ganglia.
Thank you so much for going more in detail about the perivascular spaces. And you mentioned perivascular spaces is a biomarker. How do you anticipate that our understanding would affect clinical practice in the future? Are there other biomarkers that we can use at this point to help us with diagnosis, treatment, prevention?
Yeah, great question. And there's been some impact on clinical practice already. So the validated research criteria for diagnosing cerebral amyloid angiopathy are called the Boston criteria and were recently revised to a version 2.0. The prior versions of the criteria relied heavily on hemorrhagic markers in the brain to make an in vivo diagnosis of CAA. We usually call it CAA because it's cerebral amyloid angiopathy is a bit of a mouthful, so I'll switch to calling it CAA from here on in. Prior diagnostic criteria for CAA emphasized the hemorrhagic features of the disease, where you could say that CAA is probably present based on a pattern of multiple hemorrhages in the lobar brain regions or in the subarachnoid space or the convexity over the hemispheres. The new criteria 2.0 add to that new white matter features, one of which is a moderate or severe number of visible perivascular spaces on T2 weighted or T1 weighted imaging. So it's enhanced the sensitivity of the criteria without sacrificing the specificity. That's an important advance in improving the accuracy of diagnosing CAA, and the new criteria have been validated against neuropathology from hematoma resection specimens, biopsies, or full autopsy. So that's one way that being aware of perivascular spaces has, influenced practice already by improving our ability to diagnose CAA during life. There are a lot of potential future ways that, like understanding perivascular space function and function of the neurovasculome, might help us improve practice in the future, particularly if it leads to an understanding of additional diagnostic markers or perhaps pathways that include druggable targets to form the basis of future therapeutics.
Thank you very much. Always interested in what advice can you give to young faculty interested in vascular neurology, but also in neurocognitive disorders, how to best set them up for studying that interplay, since it seems like the pathway ahead is not to have necessarily separate thinking into vascular biology and the brain, but rather a combined approach.
Yeah, I think you're exactly right that often the most innovation comes at the intersection of established disciplines, and that's where the growth happens. So I do half stroke, but also do half of my work in a dementia clinic. And I think that's been very fruitful for me in terms of my approach, particularly to the neurodegenerative conditions and for identifying causes of cognitive decline. But I think having more people like that and not being afraid to be kind of at the border zone between two different disciplines, sometimes it's you feel a little bit tugged by either discipline and you kind of lament that you're kind of stuck in the middle. But I think that's where the innovation often occurs. I think for younger clinician scientists too, I would say I'm very excited about the kind of tools that are evident. Their importance will continue to be maintained and expand in both neuroimaging as well as genomics and other forms of omics technologies are really interesting and exciting. The neuroimaging, the physics is so flexible, even after decades of use of MRI, new techniques to measure things like bulk movement of fluid or vascular activity or blood flow are still evolving and being refined. So that's a real area for future research, development, and growth. And I think likewise for genomics and other omic technologies, particularly using techniques like Mendelian randomization to do more robust epidemiological studies, are going to enhance our understanding of mechanisms and point to the next generation of clinical trials in this area. the imaging and the genomics and proteomics also allow translation back and forth, I think, between the human scenario and the animal model or tissue models that our basic scientists are working on too, because we can often look at some of the same readouts in both the human and the model systems. And by going back and forth to refine our model systems to be better models and then take the discoveries and do clinical trials in humans will hopefully accelerate the pace of discovery in the future in this area.
Terrific. I want to thank you very much for speaking with us today, Eric, and for your time and expertise in taking us on that journey, understanding the neurovasculome, learning about it, and how it hopefully can help improve. It was a sincere pleasure to have you on the podcast today.
Thank you, Christoph. Yeah, I had a lot of fun. Thank you for the invitation. I really enjoyed it.
That was Dr. Eric Smith, Professor of Neurology, Radiology, and Community Health Sciences at the University of Calgary, interviewed by Dr. Christoph Stretz, Assistant Professor of Neurology at the Warren Alpert Medical School of Brown University. ANA Investigates is the official podcast of the American Neurological Association. The show's producers are members of the ANA Education Innovation Committee, chaired by Megan Ritchie. I'm Adeline Goss, podcast host and executive producer. Our program was produced today by Caitlin White, Christoph Stretz, and me, with help from Jen Hurley. You can listen to past episodes of the show, subscribe to the show so you never miss an episode, and access all of the ANA's educational programs on the Education tab at myana.org. That's it for ANA Investigates. See you next month.